In this series of lectures, we'd like to bring in the concept of linkage. And linkage is going to connect us between classic Mendelian genetics, because we're still going to look at Mendel and Mendelian ideas, and then link that to chromosomes. So we're kind of in a corridor, if you will, of major areas between what we would call transmission genetics and what we will call in the future cytological genetics, the actual study of chromosomes. But we're going to talk about linkage and how loci are carried together on these structures that we call chromosomes, and how that affects the interaction about the expression of those as far as the phenotypic outcomes and certainly the ratios. Now, we've already dealt with linkage. We've already talked about sex linkage, and we talked about the fact that there are loci being carried around on these chromosomes that affect sex. But it turns out that linkage affects every locus because they're always connected to other loci on the same chromosome. Now, just some words that we need to think about: linkage itself, and we will define that more clearly. But for our purposes and the way we're going to look at it is that it is a violation of Mendel's principle. And the principle that we're going to be violating is the principle of assortment, Mendel's second principle. So this is the first time we've talked about that. We've talked about how important these principles were. I mean, they're ideas that they're so critical that we call them principles, for one thing, which are very rare in biology. We don't have a lot of principles because things are so variable. But certainly segregation and assortment are so common and so widespread and explain so much that we call them principles. And now I'm going to turn around and violate one of those principles and talk about the outcomes and why that might occur. We'll talk about two types of linkage, one called complete and one called incomplete. Complete will set the boundaries for us as the violation of Mendel, and then incomplete will fall in the middle, and that's where reality occurs most of the time. We'll talk about crossing over and recombination, because those are the things that move us back toward Mendelian outcomes. And there's, unfortunately, you've probably up to this point had these words almost simultaneously used, crossing over and recombination. They're not the same thing. Crossing over is as a physical effect, physical activity of the chromosomes themselves. Recombination is genetically what can happen from crossing over. You don't have to have it occur from crossing over, but sometimes when you have crossing over, or many times, you'll have recombination. And we'll talk about the physical unit where this occurs, called the Synaptonemal Complex, which you probably remember from our discussion about meiosis and mitosis, but particularly meiosis, where we talked about the formation of these tetrads, and that formation is called synapse, right? And that whole structure is called a synaptonemal complex. That's where a recombination is going to occur. Then we could have things called linkage groups, so we'll discuss and we'll talk about linkage maps, and very importantly, these things called map units, which are ways of measuring the distance between loci long before we could physically go measure the distance in a chromosome. But we could guesstimate the distance, and the same as in miles or kilometers or feet, based on some things that we can do relative to simple crosses. All right, so what I thought we'd do is just go kind of bring us back to what things look like, and assure you that we're not going to get very far away from basic Mendelian outcomes and certainly Mendelian expectations. So here's a little thing from our text, and it talks about a very simple system where purple flowers are dominant over red, and long stemmed or long pollen is dominant over round. Okay, simple dominance. We're not worried about epistasis or incomplete dominance or anything or lethal. So we just got a simple system where it's a dihybrid, and for one trait you've got a dominance going on, and for the second trait you have Mendelian dominance going on as well. If we have two true breeding homozygous strains of purple longs and red rounds, and we could breed these together, of course they'd be true breeding. These would be true breeding. So they're classic parentals. If we're under Mendel, there's a basic expectation of what might occur, right? If we were going under Mendel, we would say that one of them is a homozygote for the one, the other is the homozygote for the recessive. If we cross those, we expect to produce an F1, where we get a dihybrid het, and the dominant one for the A locus will dominate, which happens to be purple, and the dominant for the B locus, which happens to be long, will dominate. You'll end up with everybody being purple long, classic Mendelian. Then if we take those and we use simple Mendelian segregation, right? So assortment allows us to separate them. Segregation allows us to calculate the 3-to-1 ratios. We would expect some 9, 3, 3, 1 when we rebuild these ratios as phenotypic ratios. And that's exactly what we get. This is all, old hat for us now. It's nothing that you wouldn't automatically do almost by hand just instantly. But we would expect a 9, 3, 3, 1 ratio, okay? Well, let's see if that's what we get. The cross is made. These are true breeding again. So the cross is made between A and B, and we expect C to be all purple flowers with long pollen. Sure enough, they all are. Everything's perfect there. It's following a classic Mendelian. Now, if we take these, and because they're plants, we can self them, right? They'll self-fertilize. In this particular case, if we could do that, or if we put them up with, flowers of the same genotype, it wouldn't matter, but we'll just self these. Or are we going to get our 9, 3, 3, 1? And it turns out that we certainly get everything we expect. We get some purple longs, some purple rounds, some red longs, and some red rounds. But that certainly doesn't look like a 9, 3, 3, 1 ratio to me. It looks very different. Well, and that's true, and it is very different. In fact, if we can prove that by doing a classic chi-squared test, where we have the 3 genotypes, our expectation is a 9, 3, 3, 1 ratio. We have these observed data. This totals up to 381, if I did my math correctly, and you should always check me. All right, so 9/16, 16 parts, 9/16 of 381 over here should give us about expectation around 214. I put approximately because there's some decimal points there, about 71 round purple rounds, about 71 green longs, because that's 3/16, 3/16. And then 1/16 should only give us 23, yet we got 55. So we didn't get near as many of these as we expected, okay? We got way too many of these, and we didn't get near enough of these. In fact, we only got about a third of what we actually expected in these intergredient groups. So, of course, if you do a chi-squared, you've got an observed minus expected squared and all that, you get a huge observed chi-square, 134.7. We only expected it to be a table value of about 7.81. So we would reject the idea that this is 9331. It certainly doesn't seem to be a 9331. So what's going on? Well, this is a case of linkage. Turns out that the loci that are controlling the flower color and the pollen shape are located on the same chromosome. So you may remember way back when we first talked about what a gene was and all that, we said, okay, you've got this chromosome thing, and if you grab both ends of it and you stretched it out, you would have a chromosome, and we talked about a locus. And then at the same time, I said, oh, by the way, because this is one long piece of DNA, and it's many thousands, sometimes millions of bases long, you're going to have multiple genes found there. And we talked about those as loci. Now, up to this point, we haven't worried about those loci being on the same chromosome, except for sex linkage. But for everything else, we've assumed anytime you had a dihybrid, that locus A was off on one set of chromosome pair, number 2 or 3, and B was off on 12 or 13 or 20, right? They didn't travel together. All right, what we have now is we have linkage. All right, to define linkage, we would say there are many definitions, but I think this covers most of it. It's the situation, condition in which 2 or more loci tend to be inherited together, okay? That's the way it works. 2 or more loci, so they tend to travel as units. Particularly, what we see are certain phenotypes of those loci being inherited together. But the reason is that they do that is that physically the loci travel together on the same chromosome. Okay. There are 3 basic things we can say about linked genes. First is, by definition, they have their loci on the same chromosome. Secondly, depending on where they are, they tend to fail to assort independently, okay? So if they're very close together on the same chromosome, like this, they're going to fail to assort independently. Turns out that if they get very far apart on that chromosome, opposite ends or something, they actually will look to us like independent assortment, but in reality, they're actually violating independent assortment. And we're talking about why. But basically what's going to happen, the big but here, is that the reason they kind of can look like independent assortment is that if you're allowed to have crossing over and then recombination, you can get what looks like independent assortment. In fact, in Mendel's P's, he got down to, you may remember, he got down to 7 traits, right? and that he could understand, 7 loci. Well, it turns out that that peas that he works with have 7 pairs of chromosomes, but he wasn't lucky enough to get each trait on a separate chromosome. It was actually on 4 chromosomes, so he had linkage. But because they were so far apart, they didn't look like they were linked. They looked like they were independent. Fortunately for us, or we would maybe be set back even further trying to understand it. And just put it in perspective, fruit flies, we know they have 4 pairs of chromosomes, a sex chromosome and 3 autosomal pairs. They've got at least 15 to 17,000 genes that make up the entire life stages and functioning of a fruit fly. So you just divide 17 by 4, you're seeing that there's got to be lots, 17,000 by 4, there's got to be lots of loci on a given chromosome. Now humans the same way, right? We've got somewhere between 21, 22, 23,000 actual functional genes that we have, plus lots of other things that we'll talk about later. These are divided into 23 pairs of chromosomes, so we won't tend to have quite as many genes per chromosome as maybe fruit flies. But it turns out it's not even that smooth. You can't just divide 21,000 by 21 or 22 or 23 and say, oh, there's about 1,000 per chromosome. It doesn't work that way. Turns out that as we'll explain later, our genes are actually in sort of dense areas on chromosomes, and then there's not as many genes, and then there's a bunch of dense ones. It's kind of like, I say, like when I'm in a small plane, when I'm flying over an area, maybe in Texas or someplace or any place, and look down and there's a city or town, lots of people, and then they may not be very many for a while, maybe out in some open countryside, and then you're back over a city, and there's things. Well, our chromosomes, our genes on our chromosome, our loci, are basically somewhat like that. Okay. All right, now we need to get a few terms down. That's part of this session is terminology. So let's talk about crossing over. Crossing over is the exchange of genetic material between 2 homologous chromosomes by physical breaking and uniting of the material. And there's whole bounds of at the molecular level of all this enzymatic activity that goes on with these chromosomes that actually the crossing over and the recombining through these polymerases and things is a pretty cool idea. We're not going to dig through that. We'll talk about that later in either biochemistry or cell, but you do the whole process is interesting. Now, when you have crossing over, you may have recombination. Okay. Now you don't have to, okay, but you may have it. Now, what the recombination has 2 important parts: unique genetic material between non-sister chromatids. It occurs during meiosis. So unique genetic material. It means that whatever you originally got from mom, maybe you got big A and big B, and you got little a, little b from dad. If you somehow cross that over so that you get big A, little b, something, you've exchanged unique genetic material, and it's between non-sisters, right? Okay, remember sisters are these replicated chromosomes right here. Okay, but recombination is required. Now, what happens is you have to have crossover to have recombination, unless you've got mobile DNA or something. But we're talking about in normal situations, you'd have to have crossing over in order to have recombination. You can't have recombination without crossing over. Now it turns out that you can actually cross over between sister chromatids, right? Sister chromatids cross over. You may remember a picture like this back when we talked about replication, and we talked about it being evidence for semi-conservative replication. And I said, oh, by the way, there's some little places here where you've made new DNA, but you notice that there's these little hot spots. Remember these little dots are where there's radioactivity from the old strand, and then you got the new strand. And I said, look, they shouldn't be over here, but they in fact are. And people got confused by that, and they thought that was why dispersive replication might be an idea. But in fact, what that represents is that these 2 sister chromatids. So you had these chromatids, you made sisters, and somehow these sisters crossed over. Now that's crossing over, but that's not recombination, because a sister is a perfect molecular copy of the other, right? So if you've got big A here, you got big A there, when you exchange them, you still just have big A on both. You didn't do any recombination. To have recombination, it has to occur between the non-sister chromatids. So it has to occur between these guys, okay? So they're in the duplicated state. You form a synaptonemal complex, and then any of these guys can interact. They usually show it in the middle like this, but in fact, this is sort of it's a three-dimensional figure where they're all piled up, and so they can cross in all sorts of ways, and they will exchange information. If you get an exchange, in this case, showing a classic case of true recombination, where one homolog is big A, big B. Of course, its sister's big A, big B. The other homolog from the other parent was little a, little b. When you exchange, you end up with big A, big B. That's this one that didn't combine out here, shown on the outside, of course. You end up with the one that did not recombine down here. That's this other homolog. So we'll pretend that these are the outer side ones and they didn't recombine. And then you've got the middle ones that actually recombined. You had crossover and recombination. So now one of them, when they pull apart, has got a big A and a little b. And this one's got a little a and a big B, right? And so that's what you see in the middle. So when you have this sort of crossover, you end up with two parental-like, as they're called, non-crossovers, and then two that in fact show up looking like what we're going to call recombinants. Okay. All right. Now from that, since there are lots of genes on a chromosome, right? Lots of loci. These are called linkage groups, and these are groups of loci located on the same chromosome. And from that, you can develop what are known as linkage maps. These are little graphical representations of where loci are found. Are they found near the centromere? Are they found up near the top or the bottom? And where are they? And how far apart are they from the other loci that are found there? And that how far apart they are is what we call map units. Okay. And we're going to use a classic definition in here. There's more modern ways of doing it, but this will work perfectly to try to understand it. One map unit is equal to 1% recombination. Okay, so one map unit, 1% recombination. So this estimates how far they are. If two loci are really close together, then the map units, they won't recombine very often because the chance of breaking in between them is pretty low. So their map units will be very short, maybe 2, 3, 5 map units. If they're a long way apart, you expect a lot of crossover to go on. They might be 20, 30, 40, 50 map units apart. Okay. That's one of the reasons that when you're completely down at the other end of the chromosome from the top, you almost always get a crossover. So it looks like independent assortment anyway. Okay, but we're talking about one map unit is 1% recombination. Write that down, put a big star by it in your notes because we'll come back to it over and over again, and people just they forget what that is. If you forget what it is, it's going to be difficult. All right, well, let's look at one of these linkage maps for a moment. This is a classic linkage map. Okay, this is human chromosome 19. This is the short arm and the centromere somewhere here, the long arm as it might appear. And these are just, in this case, disease-related genes. So they've been mapped to some area on these particular chromosomes that we have. If you go and there are ways to actually go and back to my pointer if I can here. That's my only point. There are ways to actually go and find every human chromosome that's out there, every arm, every disease, and you can do that by simply going to the National Institutes of Health and searching for human chromosome markers, and they'll give you these patterns, and you click on it and you can find out all the thousands of genes that are on chromosome 1, 2, 3, 4, all the way up to X or Y. Okay. Now, one of the cool things that's out there is that there are connectors to show you that there's between particularly mammals and other things, there's going to be a relationship between what's found on one chromosome and what's found on the one on the other species. For example, this area of chromosome 19 in humans is found on around chromosome 10. Same genetic information is found on chromosome 10 of a mouse, of a lab mouse. Okay, this whole section right here that's found on chromosome 19 of humans, almost all of that is found on chromosome 7 of mice. And then you see that some of these others are spread around chromosome 17. So, we have the same kind of mammals. We have some of the very similar types of genes. We just reparceled them into different areas of the things as we made up different chromosome types over time. But it's kind of a nice link to see that. And this is one from, chimpanzees, or as we say, are 98% U, that we share a lot of commonality. And these are just patterns of chimp chromosome 1 versus human. So H is human, C is chimp. And these show you all the places that they share commonalities of the same exact genes or whatever. They may have mutations in them so that they're not exactly the same code, of course. But as far as the number of genes and where they're located, there's a huge number of similarity between chimpanzee chromosomes and humans, as you'd expect from both being from the simian line, from both being from the great apes and all that. Okay. All right. Now, you got all that. Why in the world are we putting all this through all this besides learning a little bit about linkage and stuff? Well, there's a number of reasons to kind of study these patterns. One is that there's chromosome location studies, right? Back before we could sequence entire genomes and figure it out, we would try to figure out where this particular location of this chromosome, where this trait was found, the gene was found, and we do that by what are known as location studies. We'll talk more about those in a minute, and you're actually going to practice some of those in labs. By studying the structure of the chromosome and where they're found, we can understand what happens when we have what are called chromosomal mutations. Now, so far with most of Mendel, we've talked about what are called point mutations, where we just change a single base, which eventually changes some amino acid or something in the protein. But what we're talking about here is that we may in fact lose large chunks of a chromosome. Well, when you do, what do you lose, right? Or what do you gain? We can actually gain large chunks. What genes are there for that to happen? And this leads, of course, to the study of things like genetic diseases and understanding just chromosomal changes and what genes are involved if I lose a piece of this chromosome number 6 right here or something. What does that relate to genetic disease? What parts are there? Okay, and then ultimately we're really interested in understanding gene regulation. So not only are we interested in figuring out where the locus is found on the chromosome, okay, but what's controlling that? There are other pieces that are there. So we may have these these 21,000 genes, but there's also thousands and thousands, many more thousands control elements that go with each of those genes. In fact, we pretty much know that there's about at least 100,000 and probably 120,000 proteins that make up a human system, right, that are required throughout our lifetime at various places in various organs and tissues. There's only 20,000 genes. You got 100,000 proteins. A gene makes a protein. So what's the connection there? And how do you regulate where those proteins are active and what forms are active, et cetera? All come down to mapping. So we don't just necessarily map loci. We're now mapping a lot of this gene regulation material. All right, just to kind of give you a handle on what you're going to be doing in lab, though, you will be doing both lab and lecture what's known as 2-point mapping. That's what we'll be concentrating on problems for the exam and problems from your textbook. 2-point map are just basically figuring out the location on a chromosome by simply doing Mendelian dihybrid crosses, but you can also figure out distance. If you know the location of one, you can use, and it's on chromosome 2, you can use 2-point mapping to say, oh, I know the other one is close by, it's 10 map units away, so I can figure out the distance. A more sophisticated way to do this is what we're going to talk about in lab, which is 3-point mapping. 3-point mapping, we take 3 loci, usually 2 of which we know where they're located. We know exactly where the map units are, and then we can figure out the distance of the third locus and this sort of thing. So it tells us the distance. It tells us whether locus 2 is in between 1 and 3, or whether locus 2 is off down here somewhere. So that's called the order of the loci. And it's really important because it will allow us to figure out distances that are greater than 50 map units. If you just have 2 loci that you're studying and they get to be above 50 map units apart, well then because a map unit is 1% recombination, right, percent, then it looks like independent assortment. So you can't really tell what's going on. This way we can map this one to be 40 and this one to be 50 from there. So we know that from here to here is 90, which we could never map locus 1 and 3 by just doing 2-point mapping. We'd have to use something that's intermediate with a 3-point. So we will work with that. That again, primarily you'll be doing those problems in lab, and we'll be concentrating mostly on 2-point mapping, or at least 2-point types of linkage up here. All right, so that's a general overview. In part 2, I want to come back and I want to talk about one of the forms of linkage, complete linkage, and introduce another concept known as configuration, which is going to be very critical in helping you understand some of these linkage problems.